Today, our mission is to completely demystify the microscopic economy of the human body. We're looking at a comprehensive textbook chapter on the fundamentals of human energy transfer.
Which sounds dense, but it's actually fascinating.
It is, because we want to know, how do you actually take, like, a turkey sandwich and turn it into the mechanical force required to run for that train?
Right. And to understand what's happening inside your muscle cells in those first few seconds, we kind of have to strip away the colloquial ways we talk about energy.
Okay, let's unpack this.
Yeah, so you have to look at the first law of thermodynamics.
Right. Physics 101.
Exactly. The human body does not produce energy. And it doesn't consume energy either. Well, not in the strictly physical sense.
Because energy can't be created or destroyed.
You got it. It's entirely a system of energy transformation. The source text we're using frames this whole biochemical reality around this really ancient, elegant visual, the water wheel.
Oh, I love this analogy. So if we think about a hydroelectric dam, right?
Yeah.
You have this massive reservoir of water just sitting quietly at the crest.
Completely still.
Right. And that is potential energy. It's trapped, it's structural, it's just waiting there.
Waiting to be released.
Exactly. And then when the floodgates open and that water cascades over the dam, it transforms into kinetic energy, the energy of motion.
Right. And if you place a water wheel or like a modern turbine into that falling water, you harness that kinetic flow to perform useful mechanical work.
So how does that map onto the turkey sandwich?
Well, the food you consume, the carbohydrates, the lipids, the proteins, that is the water sitting at the top of the dam.
Okay, so the macronutrients are just packed with potential energy.
Exactly. Chemical energy stored right within their molecular bonds. And human metabolism is basically the biological water wheel.
Got it.
The body systematically breaks those chemical bonds, which releases that stored potential energy, and then transforms it into kinetic energy.
And that kinetic flow powers the biological work.
Exactly. It powers the mechanical work of your muscle fibers contracting. It powers the chemical work of synthesizing new cellular tissue.
Even brain function, right?
Oh, absolutely. The transport work of pumping ions across cellular membranes so your neurons can fire. It is all just energy flowing downhill, carefully channeled and harnessed.
Okay, but we know the food is the potential energy. But a muscle cell can't just, like, take a literal piece of bread, slap it onto a protein filament, and expect the fiber to twitch.
No, definitely not.
Because food is like a solid gold bar.
Oh, that's a good way to put it.
Right, like, it holds an immense amount of value. But if you go to a cafe and try to buy a coffee with a gold bar, the transaction fails.
They're not taking that.
Right. Your cells only accept cash. So we have to look at how the body breaks down the gold bar of that sandwich into spendable biological money.
And that universal energy currency of the cell is adenosine triphosphate, or, as most people know it, ATP.
ATP, the biological cash.
Exactly. It was discovered back in 1929 by a guy named Carl Lohmann, and he realized that this specific molecule is the absolute prerequisite for all cellular functions.
Without it, nothing happens.
Nothing. Without ATP, everything just stops. So structurally, you have an adenosine molecule linked to 3 phosphate groups.
Hence the triphosphate.
Right. And the bonds holding those outermost phosphates are highly unstable.
And the actual spending of that currency is like a really violent chemical reaction.
Oh, extremely violent. The cell performs a process called hydrolysis.
Which means using water to break it.
Exactly. It uses water to just cleave that outermost phosphate bond right off the ATP molecule.
But wait, how does snipping off a single phosphate group actually provide the physical force to move a muscle?
It basically comes down to electrostatic repulsion.
Okay.
Those 3 phosphate groups, they're all negatively charged.
And like charges repel.
Exactly. Because they have the same charge, they naturally want to push away from each other, but they are chemically forced together in this molecule.
So it's like compressing a heavy coiled spring and just like locking it in a tiny box?
That is exactly what it is. When the enzyme snips that 3rd phosphate bond, it's releasing the latch on that compressed spring.
Wow.
The phosphate flies off, and that sudden physical release of tension liberates a specific, highly useful amount of free energy.
How much energy are we talking about?
Splitting 1 mole of ATP yields about 7.3 kilocalories of free energy.
Okay.
And that little explosion of kinetic force is transferred directly to the muscle proteins to make them slide past one another and physically contract.
Okay, wait, I have a question about this. Because if the biological cost of movement is paid by splitting ATP, the math in the source material presents a massive problem.
Ah, yes. The storage issue.
Right. The text states that the human body only stores about 80 to 100 grams of ATP, at any given time.
It's tiny.
That is a tiny amount of currency. The text says that's only enough intramuscular stored energy for about 3 seconds of explosive all-out activity.
Roughly 3 seconds, yeah.
So if I only have 3 seconds of ATP cash on hand, I should be collapsing onto the pavement long before I ever reach that 8.15 train.
You absolutely would.
So what is keeping the system online past the 3-second mark?
Well, your body operates a hidden instantaneous backup generator.
Okay.
Because if you relied solely on stored ATP, I mean, a 100-meter sprinter wouldn't even make it out of the starting blocks.
Right.
So inside the muscle cell, co-located right next to the ATP, is a second high-energy compound. It's called phosphocreatine, or PCR.
PCR.
Yeah, PCR is the hidden reservoir. It contains its own highly volatile phosphate bond.
Okay, so it has a spring in a box, too.
Exactly. And the absolute microsecond your stored ATP starts getting depleted. So when that third phosphate is cleaved off, leaving behind a spent 2-phosphate molecule called ADP.
Adenosine diphosphate.
Right. The PCR system just activates instantly.
It essentially steps in to fund the deficit.
Instantly. PCR splits its own phosphate bond, releases its own explosion of energy, and uses that energy to violently glue a new phosphate group right back onto the spent ADP.
Oh, wow. So it just repairs it on the fly.
It resynthesizes the ATP molecule almost instantaneously. It takes the spent currency, repairs it, and hands it right back to the muscle to spend again.
That is incredible.
And it does this without needing a single molecule of oxygen.
What's fascinating here is that the body doesn't solve the energy problem by hoarding massive amounts of cash.
No, it doesn't.
It solves it through high-speed recycling. Because, I mean, if biology had engineered us to simply store all the ATP we needed for a full day of movement.
Oh, you wouldn't survive it.
Right. The sheer physical weight of those molecules would be lethal.
Yeah, a sedentary person just sitting around uses an amount of ATP every single day that is roughly equivalent to 75% of their total body mass.
That's insane. And for an athlete.
A marathon runner might generate, split, and recycle up to 80 kilograms of ATP during a 3-hour race.
80 kilograms. You literally couldn't physically carry that.
Exactly. So biology gave you a microscopic stash of ATP, a slightly larger stash of PCR, and just an ultra-rapid recycling protocol.
Okay, but even with PCR doing this rapid repair job, the reservoir is highly limited, right?
Very limited. It reaches its maximum energy yield in about 8 to 12 seconds.
So by the time you were 10 seconds into your sprint for the train,
the ATP is depleted and the PCR backup generator is completely tapped out.
And the muscle is screaming for more ATP.
But oxygen hasn't had time to travel from your lungs down to the local tissue yet.
Right. 10 seconds isn't enough time to start breathing heavy and delivering oxygen.
So the cell has to initiate an emergency override to process raw nutrients into ATP without oxygen.
Which brings us into the realm of enzymes and rapid glycolysis.
Yes, it does.
And the speed at which this emergency override operates is honestly hard to process.
It defies imagination.
Yeah, because we tend to imagine cellular chemistry as these slow, ambient reactions, like stuff just floating around in a pool.
Like a lava lamp or something.
Right. But the textbook highlights the specific physics of enzymes as biological accelerators. It points to one enzyme, carbonic anhydrase, whose job is to hydrate carbon dioxide.
Yeah.
Its turnover rate is 800,000 molecules.
And it's not per minute.
No, per second. 800,000 molecules per second.
The sheer quantum mechanics of that speed are staggering. And you have to remember, enzymes do not cause chemical reactions that wouldn't otherwise happen.
Okay, so what do?
They are specialized protein catalysts. They drastically lower the activation energy required for a reaction to occur.
So they just make it easier.
Way easier. They contort and bend the substrate molecules into such unstable, vulnerable positions that just the ambient thermal energy of the cell is enough to instantly force the reaction. So when your PCR runs out, a highly orchestrated cascade of these enzymes kicks into overdrive in the watery fluid of your cells, which is called the cytosol.
The cytosol.
Yeah. And this whole process is rapid anaerobic glycolysis.
And glycolysis literally means the splitting of sugar.
Exactly.
So the cell grabs a molecule of glucose and just rips it apart through this 10-step enzymatic cascade.
Synthesizing a small amount of ATP just to keep your legs pumping.
And crucially, it does this completely without oxygen.
Yes, anaerobic.
And this buys you up to about 90 seconds of high-intensity sprint time.
It does, but operating an emergency engine without oxygen comes with a severe structural bottleneck.
Exhaust.
Right. As those enzymes tear the glucose molecule apart, They are stripping away hydrogen atoms.
Okay.
Now, normally, those hydrogens would be fettled deep into the mitochondria to be processed smoothly using oxygen.
But because you are sprinting, the system is overwhelmed.
Oxygen just isn't present in sufficient quantities yet, so the hydrogen atoms start piling up in the cytosol.
Here's where it gets really interesting, because if those hydrogen protons accumulate, the pH of the cell drops rapidly.
It becomes highly acidic.
Right. And if the cell becomes too acidic, those lightning-fast enzymes literally warp, they change shape, and they stop working.
The muscle contraction just grinds to a painful halt.
Which brings us to arguably the most misunderstood molecule in all of exercise physiology, lactic acid.
Oh, absolutely. The villain of the fitness industry.
We culturally vilify lactic acid. We blame it for the burn. We blame it for fatigue. I mean,
Right.
we buy expensive recovery tools, massage guns, just to, quote-unquote, flush it out of our legs.
Which is completely backwards. The source material completely flips this narrative.
Thank goodness.
Yeah, it categorizes lactate as a highly valuable metabolic intermediary.
So it's not bad.
Not at all. To stop the cell from choking on its own accumulating hydrogen exhaust, the body deploys this elegant, temporary storage solution.
Okay, how does that work?
It takes a molecule called pyruvate, which is just the end product of splitting that glucose, and it uses it as a biochemical sponge.
A sponge.
Yeah, pyruvate binds with those excess acidic hydrogens to form lactate.
Oh, so it's not a poison causing the fatigue. It's the rescue vessel preventing immediate failure.
Exactly. By soaking up those hydrogens, it keeps the cell's pH stable just long enough for glycolysis to keep running.
That completely changes how I think about the burn.
And its utility goes far beyond just acting as a sponge. This introduces something called the Cori cycle.
Right, named after Gertie and Carl Cori.
Yes, the Nobel laureates who mapped it out. The lactate doesn't just sit in the muscle causing pain, it diffuses out of the muscle cell and right into the bloodstream.
Where does it go?
The vascular system carries it directly to the liver.
The liver.
Yeah. The liver is the heavy biochemical processing plant of the human body. It takes that lactate and, through a process called gluconeogenesis, strips the hydrogens back off and reassembles the fragments straight back into fresh glucose.
Wait, it makes new sugar. It's an entire closed-loop recycling program.
Exactly. The liver dumps that newly minted glucose right back into the bloodstream, where it travels back to the sprinting muscle to be split for energy all over again.
So the lactate is actually a highly mobile, circulating fuel source, keeping you alive when oxygen is scarce.
It's saving you, not hurting you.
Wow. Okay, so we have mapped the initial timeline. Stored ATP covers the first 3 seconds.
Yep.
The PCR backup generator bridges you to 10 seconds.
Right.
Rapid glycolysis and the Cori cycle push you through the 90-second mark of maximal effort.
All anaerobic.
But what happens when you finally catch the train, you sit down, and then you realize you got on the wrong line, and now you have to walk 4 miles home.
Ah, the endurance phase.
Right. To sustain muscular contraction for hours, the metabolic location has to physically migrate.
Right, it does. We must move out of the watery cytosol and deep into the inner matrix of the cell's primary power plant,
The mitochondria.
the mitochondria. We shift from anaerobic glycolysis to cellular oxidation.
This is where we bring the water wheel analogy back in full force.
Oh, definitely.
Because the source dives into the citric acid cycle and the electron transport chain. But instead of getting bogged down in the alphabetical soup of all the chemical intermediaries,
Because there are a lot of them.
yeah, too many.
Yeah.
Let's just look at the mechanical genius of the mitochondria.
Sure. So when oxygen is finally present in abundance, the fragments of your food are systematically stripped of their remaining hydrogen atoms.
Okay.
And those hydrogens are passed down a biological bucket brigade called the electron transport chain.
Which is embedded in the inner wall of the mitochondria.
Exactly. And this is where the biological dam actually operates. The electrons from your food are used to physically pump hydrogen protons across the mitochondrial membrane.
Pumping them against their will.
Right. It creates a massive, highly concentrated reservoir of protons on one side. You are literally pumping water to the top of the dam.
And because they are positively charged, these protons repel each other aggressively.
They want to get away from each other. They want to flow back across the membrane to equalize the pressure, but the membrane is impenetrable.
Except for one specific channel.
Exactly.
A channel that is literally a biological turbine.
Yes. An enzyme called ATP synthase.
The protons rush through that channel like water over a dam, right?
And the physical force of that rushing river of protons actually physically spins the enzyme.
It spins it like a real turbine.
Like a literal motor. And that mechanical spinning force provides the massive energy required to slam phosphate groups back onto ADP.
Synthesizing massive amounts of ATP.
Yes. The efficiency of this aerobic system is profound.
How profound?
Well, when you process a single molecule of glucose completely through the mitochondrial turbine using oxygen, the net yield is 32 molecules of ATP.
32.
Contrast that with the measly 2 or 3 ATP you get from the anaerobic emergency override.
Oh, wow. That is a huge difference.
It's massive.
But, and this is wild, even 32 ATP from a glucose molecule looks like loose change compared to the payout of lipid or fat metabolism.
Oh, yeah. Fat is a whole different ballgame.
Because the source heavily contrasts carbohydrate breakdown with fat breakdown. It says a single molecule of stored fat, a triacylglycerol,
Right.
is broken down into a glycerol backbone and 3 long fatty acid chains. And when those run through the mitochondria, the yield is an astonishing 460 ATP.
450-60. From one molecule.
How is that even possible?
To understand why the payout is so massive, you just have to look at the structural carbon geometry.
Okay, geometry.
A glucose molecule only has 6 carbon atoms. There are only so many hydrogen bonds you can strip away from a 6-carbon structure.
Right.
But a triacylglycerol molecule contains 3 sprawling fatty acid chains, which can be 16 to 18 carbons long each.
Oh, wow.
Yeah. So the mitochondria run a process called beta oxidation. It systematically chops those massive carbon chains into 2 carbon fragments.
Just chopping them out.
And that releases an absolute deluge of hydrogen protons to pump up the dam.
And more protons rushing through the turbine means exponentially more ATP.
Exactly, 460 of them.
If we connect this to the bigger picture, it explains the fundamental architecture of human anatomy, doesn't it?
It really does. Carbohydrates are structurally inefficient for long-term storage because they require a significant amount of water to be packed away in the tissue.
They're bulky.
Very bulky. A well-nourished, college-aged male only has about 2,000 kilocalories of total carbohydrate stored as glycogen in their liver and muscles.
2,000 calories. That's, what, only enough fuel to run about 20 miles before the tank is completely bone dry?
Right around there, yeah. But fat, however, is anhydrous.
Meaning it doesn't hold water.
Exactly. Because it is so compact and energy dense, that exact same lean individual carries up to 100,000 kilocalories of energy securely stored inside their adipose tissue.
100,000 calories.
It is a vast, nearly inexhaustible ocean of potential energy, yielding 460 ATP per molecule.
Which demands a very practical question, I think.
Let's hear it.
If a fat molecule gives us a massive 460 ATP payout, and a glucose molecule only gives us 32 ATP, why do marathon runners carbo load the night before a race?
That's the golden question.
Right. Why aren't they just eating sticks of butter on the starting line to access that massive 460 ATP lipid payout? Why the obsession with pasta and bagels if fat is the superior fuel?
That paradox reveals the single most critical biochemical limitation in all of human performance.
Okay.
The textbook sums it up in a foundational axiom of physiology, and the quote is, "Fats burn in a carbohydrate flame."
Fats burn in a carbohydrate flame. It is a beautiful phrase.
It's very poetic for a textbook.
But what is the actual mechanism? Why does a massive fat reserve require a carbohydrate spark to ignite?
It centers on the gateway into the citric acid cycle inside the mitochondria.
Okay.
Remember that beta oxidation chops the long fatty acid chains into those 2 carbon fragments? They're called acetyl-CoA.
Right.
Those fragments need to enter the citric acid cycle to be processed, but they cannot enter on their own.
They need a key.
The metabolic cycle is a locked gear. To enter, the fat fragment must physically bind with a specific resident molecule called oxaloacetate.
Oxaloacetate. So it's like the other half of a biological zipper.
Exactly.
The fat fragment can't just slide into the cycle. It has to physically interlock with oxaloacetate to form a new, larger molecule that can actually turn the metabolic wheel.
Precisely. If oxaloacetate is not present, the zipper is broken.
Wow.
The fat fragments cannot enter the cycle, they accumulate, and fat oxidation comes to a grinding halt.
Oh, F it.
And here's the biochemical catch. The primary way the body regenerates oxaloacetate is from the breakdown of carbohydrates.
Specifically from pyruvate, right? The byproduct of glycolysis.
You got it.
So if you run entirely out of carbohydrates,
If you deplete your muscle and liver glycogen stores entirely, your body stops producing pyruvate.
and without pyruvate.
The regeneration of oxaloacetate plummets.
Wow.
Without oxaloacetate, the citric acid cycle loses its resident binding partner. The cycle slows down, and your ability to process those two carbon fat fragments drops drastically.
You still have 100,000 calories of fat sitting dormant in your tissue.
But without the biochemical spark of carbohydrate breakdown to keep the cycle spinning, you cannot access that energy fast enough to sustain high-intensity physical output.
This is the physiological reality of hitting the wall.
Yes, exactly.
It's not that the athlete ran out of fat. They have tens of thousands of calories of fat remaining.
It simply ran out of the carbohydrate flame required to burn the fat.
They lost their oxaloacetate.
Yep. That is why they load up on pasta. They are trying to physically pack as much glycogen into the liver as possible to ensure the flame never goes out.
That makes perfect sense now.
Now, the body does have a dramatic fallback protocol for when carbohydrates are entirely absent.
Okay, what happens then?
If an individual is fasting, starving, or maintaining a severely restricted low-carbohydrate diet and the oxaloacetate levels crash, those acetyl-CoA fat fragments begin to bottleneck in the liver.
Because they can't enter the cycle.
Right. So the liver takes those stalled fat fragments and converts them into strong acidic metabolites called ketone bodies.
Ketones. That's a concept dominating the diet and wellness space right now.
Oh, absolutely. Everyone is talking about ketones. And this raises an important question. Ketones are a brilliant evolutionary survival mechanism.
Because they keep you alive.
They can cross the blood-brain barrier and serve as a crucial alternative fuel source for the central nervous system when glucose is completely unavailable.
If there's a catch?
The textbook is unflinching about the metabolic cost. Accumulating high levels of these strong acids significantly alters the physiological pH.
Makes you acidic again.
Right. Without adequate carbohydrate intervention, excessive reliance on ketones shifts the body into a state of ketosis.
Which people try to achieve intentionally.
They do, but while it sustains life at a low baseline, the biochemistry clearly dictates that operating at maximum aerobic power requires the seamless, simultaneous interconversion of carbohydrates.
You keep the metabolic mill spinning so fats can be oxidized efficiently.
Exactly.
So what does this all mean? If we zoom out and just track the entire bioenergetic arc of your run for the train,
It's quite a journey.
it is. We start with a tiny microscopic stash of ATP cache that violently snaps its phosphate bonds to get you off the starting line.
Yep.
When that runs dry in 3 seconds, we rely on the PCR backup generator to instantly glue the currency back together for another 10 seconds.
Instant repair.
And then, to keep accelerating, we start splitting sugar without oxygen in the watery cytosol, rapid glycolysis,
Buying you 90 seconds.
buying us 90 seconds of sprint time, while the liver brilliantly intercepts the lactate waste, runs it through the Cori cycle, and turns it back into glucose fuel.
Such an elegant recycling system.
And finally, as you settle into a long, sustained walk, we migrate deep into the mitochondrial dam. We use a small, precious spark of carbohydrates to generate oxaloacetate, which ignites and burns our massive 100,000 calorie fat reserves, spinning the ATP synthase turbine to keep us moving forward.
That's it. That's the whole system. The source explicitly refers to the human body as the ultimate metabolic mill.
The metabolic mill.
Yeah, macronutrients, fats, carbohydrates, and even proteins. They're not static materials.
Right.
They are continuously being dismantled, stripped of their hydrogens, repurposed, and interconverted through overlapping chemical cycles simply to keep your biological turbine spinning.
It is an absolute staggering marvel of engineering.
It is, but it raises a really provocative evolutionary paradox that the source material kind of forces us to confront.
Okay, what's that?
Well, we are looking at a bioenergetic system that was meticulously shaped over millions of years of hominid evolution.
Right, our ancestors.
Every enzyme, every metabolic override, the Cori cycle, the ketone fallback, it was all engineered to harness, conserve, and ruthlessly recycle energy under conditions of extreme physical scarcity and relentless physical demand.
Because getting food was hard.
The system is designed to squeeze every singular drop of kinetic force out of a solitary glucose molecule because for almost all of human history, securing that glucose was incredibly difficult. Well, knowing the sheer physics and desperation encoded into our cells, how does the modern reality of a continuous, effortless calorie surplus paired with extreme physical inactivity fundamentally clash with this ancient high-performance machinery humming inside you right now?
Wow. You are operating a microscopic engine built for a world of scarcity, but navigating a world of absolute abundance.
Exactly, a system built for a reality that mostly doesn't exist anymore.
That is definitely a thought to sit with.
It changes how you look at a sandwich, doesn't it?
It really does. Well, thank you all for joining us on this exploration of the microscopic economy of the human body.
It's been great.
Keep diving deep, and we'll see you next time.